Welcome back to NPTEL online course on Introduction to Complex Biological System. This week, I will be discussing about infectious diseases. Especially, I will discuss about virus and bacteria. And this lecture number 41, mostly I will introduce microbial world and infectious diseases. So here, I will cover the general introduction about microbes and its impact on our environment and living world, followed by a brief introduction to microbes and infectious diseases and their complex relation between microbes and human. Now, what are microbes? So, microbes, I would say, like organisms and acellular biological entities. So, as a result of that, microbes, they can be some acellular biological entities. For example, virus, virus are not cell, they are not considered as living organism. So, that is why I am including both cellular and acellular biological entities. But the major feature is that they are very small to be seen clearly by the unaided eye. So, you need some kind of I would say microscope in order to properly visualize those small organism or acellular entities. So, those are the microbes. So, now if you see organisms and biological entities studied by microbiologists. So, microbiologists are professional who studied microbes, and they can be cellular and acellular. And among cellular, we can largely classify them in three major categories. One is bacteria, the other one is archaea, and the last one is eukarya. So, basically if we consider like prokaryotes and eukaryotes, this bacteria and archaea, they will come under prokaryotes, and then the other one is eukaryotes. So, now this classification, like here this is based on RNA sequence, the ribosomal RNA sequence analysis, and this is like universal phylogenetic tree here. So, here also another important aspect here, you will be noticing that archaea are not directly related to bacteria, like they have huge difference in terms of this classification or in terms of this phylogenetic tree. If you notice, like they have some kind of similarities with eukaryote also in terms of RNA sequences. So, anyways, so if we would like to concentrate on microbes, now here I would say this can be prokaryote and eukaryote, the way I just mentioned. The other way you can say it's unicellular and multicellular organisms. So, unicellular means here both will come, like bacteria, for example, E. coli, Staphylococcus, all bacteria will come as unicellular microbes, and other one is archaea. So, here you can say methanogens. And now multicellular organism, they can also come under microbes, because there are many multicellular organisms, they are very small. Just one example, if I say bread mold. Sometime if we keep bread for long time in moist condition, you will be noticing some whitish filamentous stuff coming up. So, those are like bread molds. So, there we have many cells there in those fungus, but still you need microscope to properly visualize them. So as a result of that, these cellular microbes can be unicellular as well as multicellular. And now acellular entities. So here the major two classes, I would say, one is viruses. As you know that viruses, they don't have their lot of machineries are missing there. So they are only replicating inside the host cell. So as a result of that, we are not considering virus as living organism. They just have like some kind of nucleic acid. It can be DNA or it can be RNA. So RNA can be genetic material for viruses, and some proteins. Very simple structure of virus particle. For example, poliovirus, just one example, and influenza virus. There are many viruses. And now another major category of acellular microbes is prion. So prions are actually protein, no nucleic acid there. So they are just protein particle. But the thing is, these protein particles are infectious. So what happened? This protein, somehow their folding and their character has been changed due to various different types of reason. And now those protein can convert other protein into a misfolded state, and they can actually make other protein into prions. So, and they are actually causing diseases. For example, they causes spongiform encephalopathies. So, for example, scrapie, mad cow disease, mad cow diseases. I am little bit elaborating about prion because prion I am not going to discuss in more details in the next class, but I will be discussing about viruses and bacteria in great details in the following lectures. So this is prion. So, and all together they are microbes. And now let us see when they evolve in terms of the evolution of earth, these microbes. So although we considered that microbial world, microbes, they evolved like at first, and then this complex organism like multicellular organism, eukaryote, we evolved much later. Let's see. So here, if you see, this is the timeline. So I would say that approximately 4.5 billion years ago. So BYA, as you can see, it's mentioned BYA, billion years ago. So 4.5 billion years ago, Earth formed, right? Now, in this time scale, if you see the origin of different types of organism, you will be noticing that 3.5 billion years ago, first cells appeared. What kind of cells? Those are nothing but prokaryotic cells, some kind of bacteria. They don't have proper nucleus. As you can see, this is the DNA, and I would say this is just prokaryotic cell. It can be bacteria or archaea. Now, again, it took long time, and approximately 2.5 to 2 billion years ago, eukaryotic cell developed. So this is eukaryotic cell, but they are still single cell eukaryotes, right? So as you can see here, this is nucleus, and here mitochondria, and this is chloroplast. And it took long time to get the multicellular eukaryote. Approximately 1.5 billion years ago, as you can see here, this is multicellular primitive eukaryotes, not like proper differentiated organism like us, like human or big plant, not like that. And I should mention here, it took many billions of years, and finally I would say approximately 2 million years ago, not billion, remember million years ago, Homo sapiens, human, right? Homo sapiens developed. So now, so as a result of that, what I am trying to say, at the beginning, there were only microbes, only unicellular organism. Now what we see, we have different types of organism, lot of diversities here, like from microscopic organism like virus, bacteria, to very complex organism like human, plant, and so many organism present in present world. Here, in order to support whatever this time scale I am just discussing, here if you see this microfossil which were found in apex chart of Australia, this is around 3.5 billion years old, and these microfossils are very similar to modern filamentous cyanobacteria. So cyanobacteria is also present now, so they are photosynthetic bacteria, but whatever found in this apex chart of Australia, this is not true cyanobacteria what is present now, but this is very close to that. So as you can see, this is found in this microfossil. So again, it suggests, if you see the time scale, that 3.5 billion years old. So as a result of that, we mention here that 3.5 billion years back first cell appeared, and here 3.5 billion years ago fossil of primitive filamentous microbes originated. Now I would like to emphasize that this early cellular life, that means both archaea and bacteria, they shape our environment and drives evolution of complex life forms. I would like to explain this part, that if you see, early Earth was a hot environment that lacked oxygen. So as a result of that, if you see nowadays, like in present world, almost all of us, we rely on oxygen for energy metabolism, right? Without oxygen, we cannot survive. But when those organisms evolved at the beginning, I would say very beginning when Earth formed, during that time, no oxygen there, and it was very hot. So currently there are heat-loving archaea. So archaea sometimes considered as more primitive than bacteria. So they are capable of using inorganic molecules such as iron sulfide as a source of energy. Instead of oxygen, oxygen was not there at that time, so they are using iron sulfide as energy source, which is more likely a remnant of the first form of energy metabolism. And over the time, oxygen generated and that also drives evolution of different types of organism. So here, that's why I mentioned that another metabolic strategy is oxygen releasing photosynthesis, and it appears to have evolved as early as 2.5 billion years ago. So as you can see here, this is like some kind of section of fossilized stromatolite. So what happened here? Fossils of cyanobacteria, this is already I mentioned that photosynthetic bacteria. So they are bacteria, but they can carry out photosynthesis. So here I am just clarifying that, as you know, that photosynthetic organism like plant, they have chloroplast, right? And there are a lot of pigments inside the chloroplast, and that's why they can carry out photosynthesis. But these cyanobacteria, they don't have chloroplast, but they have some kind of pigments which can carry out photosynthesis. And those cyanobacteria, they are found in rocks like stromatolites, and dating to that time, like around, I would say, 2.5 billion years ago. So that support this hypothesis that during that time they like generated oxygen because of these different types of metabolic activities. And the oxygen they release is thought to have altered Earth's atmosphere to its current oxygen-rich state. As I already mentioned, that now almost all of us, almost all organisms, they are relying on oxygen. Beside their role in shaping our environment, they also drive evolution of complex life forms. Like at the beginning, we had only unicellular organism, and particularly I would say prokaryote like archaea and bacteria. But then eukaryotic cell developed, and then multicellular organism. But now if we see how eukaryotic cells developed, so there are many hypotheses, and endosymbiotic origin is one of the most important, and scientists really believe this hypothesis in order to support the origin of eukaryotic cell. What happened? Like primitive eukaryotic cell, say for example some kind of big bacteria, I would say archaea. So if this is archaea, so they phagocytose small bacteria. So as a result of that, you will be saying that this is the bacteria or archaea, it can be both, and this is some kind of ancient eukaryote, not yet developed eukaryote, and the bacteria came inside this ancient eukaryote. And now over the time, it took long time, many million years, billion years time. So then today's complex eukaryotic cell developed. So as a result of that, the development of nucleus and all those more and more complexities developed. But now if you see, like this is some kind of symbiotic relation. The bigger cell, whether it is archaea or bacteria, that doesn't matter, but the bigger cell, what I am referring that ancient eukaryotic cell, so that actually phagocytose this bacteria, and now they are on a symbiotic relation, and over the time this modern eukaryotic cell developed, and where these bacteria inside these cells, they are still present, like mitochondria and chloroplast. So they are still present. So as a result of that, if you see mitochondria and chloroplast, they contain DNA and ribosome. So although they are cell organelle, but they do have their own DNA and ribosome. And more importantly, both are similar to bacterial DNA and bacterial ribosome. They are more similar to bacterial DNA and bacterial ribosome. And particularly, I would like to mention that the DNA of mitochondria and chloroplast, that is circular DNA, which is again similar to bacterial DNA. It's circular DNA. And also, both organelles belong to the bacterial lineage based on ribosomal RNA analysis also. And another important thing is the replication of these two organelles, that is mitochondria and chloroplast, is just like bacterial replication, like binary fission. That means one bacteria here, and again it's just divided into two. Similarly, chloroplast and mitochondria, they undergo binary fission and they divide in this way. So all these support this endosymbiotic origin of eukaryotic cell. And more importantly, the genome sequence of this bacteria, Rickettsia, this is an obligate intracellular bacterium that causes typhus. It is a disease, particularly fever, headache and rash. Those are the common symptoms of this epidemic typhus. And if you see, the genome sequence of this bacteria is more similar to that of modern mitochondrial genome sequence than to any other bacterium. This is very interesting. So it again suggests that this mitochondria might have evolved from bacteria itself, and that's why you can see a lot of similarity between the mitochondrial genomes and the genome of this bacteria Rickettsia. And now I would just like to focus on microbial life under microscope. Whatever, like, in modern day, currently we are studying about, microbes. We need powerful microscope also in order to understand microbes in a better way. But at the beginning, it's like, I would say long time back, it's 1665. During that time, this mold sporangia drawn by Robert Hooke. And his observation of this sporangia is based on, like, whatever he saw under microscope. So this is although just hand drawing, right? Not the microscopic image. This is just drawing by Robert Hooke, who also, discovered cell and like that cell is unit of life. Almost all of you know about that. So now Robert Hooke first draw this picture about this mold sporangia, and almost at the same time, Antony van Leeuwenhoek, he is best known for his pioneering work in microscopy and for his contribution toward the establishment of modern microbiology or microbiology as a scientific discipline. He first observed bacteria through microscope. So Robert Hooke first observed cell under microscope. So for example, mold sporangia. So as a result of that, this is not bacteria. This is some kind of eukaryotic cell, right? Bread mold, for example, it's an eukaryotic cell. But here, Antony van Leeuwenhoek, he first observed bacteria under microscope. And then long way to go. Now we have lot of information, lot of understanding about bacteria, viruses, and so many things. Now I would like to mention few of the important discovery that I would say that those are the major pillars, and that actually that are the foundation of modern microbiology. Like first one is, like, there are many. I will, like, I cannot cover all those things, but some important point I will be covering here. Robert Koch, so he devised technique of pure culture to study a single species of microbe in isolation. Nowadays, almost in all lab, like whenever we are working with some microbes, we work on a single species of that microbe, particular bacteria, for example, and in isolation, not in environment, right? So as a result of that, Robert Koch actually first discovered this technique of culturing single species. And then Koch's postulates also provide a set of criteria to establish a causative link between an infectious agent and disease. This is very important, that a particular microbe can cause a particular disease. This link, that causative link between a microbe and the disease, it was also established by Koch, Robert Koch. And then this is again another a milestone, that Edward Jenner established the practice of vaccination. So what he did, like, he inoculated cowpox to prevent smallpox. As you know, smallpox is a very deadly disease during that time, but cowpox is kind of again caused by some kind of viruses. And what he did, if someone is inoculated with cowpox, and it can prevent the smallpox disease. So this is again a great discovery, and this is modern day, whatever we are taking vaccine, immunization, the basic study coming from here, the first one. And then Louis Pasteur developed the first vaccine based on attenuated strains, such as rabies vaccine. Nowadays, like, many attenuated strain of microbes are available for vaccination. And Alexander Fleming discovered that penicillium, so this is particularly Penicillium notatum. So this is some fungus, right? So it generates a substance that kills bacteria. This is very important discovery. This is the beginning of the discovery of antibiotics. So then over the time, Flore and Boris Chen, so they purified the substance penicillin from this fungus, and this is the first commercial antibiotic to save human lives. Now we have a number of antibiotics available, but this is the beginning of that. And because of their beautiful work, all three of them, they shared Nobel Prize in Physiology and Medicine in 1945. So penicillin was the first antibiotic which was used for like to save human lives. So I'm just giving like one example, like penicillin, right? So now if I say ampicillin and then amoxicillin, so all those antibiotics, they are kind of very similar antibiotics. So penicillin was the first one, the natural one, and then over the time scientists developed little bit of different antibiotic, but all these three antibiotics, they work in the same way. So they actually block the synthesis of peptidoglycan. So peptidoglycan is present, it's a polymer, it's present in bacterial cell wall. So as a result of that, if those antibiotics, they stop the synthesis of peptidoglycan, so as a result of that, bacteria cannot make its peptidoglycan. So bacteria will die, it will not survive, it cannot divide. So that's why this antibiotic is very important. And here when I was discussing about archaea and bacteria, I must mention during that time I forgot, like one of the major difference between archaea and bacteria is, in case of bacteria, they have peptidoglycan in their cell wall, but archaea, they don't have peptidoglycan. And now I would like to briefly introduce a few microbial infectious diseases. So microbes cause diseases, and some of these diseases have had a huge impact on human history. So I will just mention few of them. If you see, in 1347, the plague, which is also known as Black Death, struck Europe with brutal force, and it killed 1/3 of the population, that is almost about 25 million people within 4 years, from 1347 to 1351. So this is a huge, I would say, huge load, huge burden, I would say. And later on, the Black Death, it was discovered that the Black Death was caused by the bacterium Yersinia pestis. So this is the bacteria, the causative organism for this disease, which is typically transmitted to human through the bite of an infected flea which had bitten a rodent, for example rat, that carrying this bacteria. But nowadays this kind of diseases like bacterial diseases can be managed much better way, because as I already mentioned, there are a spectrum of antibiotics available commercially nowadays. So it's much easy to handle this kind of bacterial infection. And then another important infectious disease outbreak was 1918 flu pandemic. So in the first lecture, first module, when I discussed about the Griffith's experiment, that was in the backdrop of this disease, that is 1918 flu. It is also known as Spanish flu, the same thing, right? Spanish flu. This is a deadly influenza pandemic that killed millions of people worldwide. And then cholera, a severe diarrheal illness caused by the Vibrio cholerae, and cholera is a waterborne disease. So here I must mention that like long time back, I would say, because of cholera many people used to die. And one of the major problem was, like in case of cholera, and it is the symptoms is severe diarrhea. So as a result of that, people were suffering from dehydration and people lose lot of water. So as a result of that, it was very difficult to manage, and it was like one of the pioneering scientists in this field. I must mention here, he is Dr. Shambhunath De. So he used to work in Kolkata, and his work particularly demonstrated that the pathology of cholera disease, that during cholera this dehydration is happening, that directly helped to the development of this ORT therapy, that means the oral rehydration therapy, oral rehydration therapy. Why? As I already told, that the pathology is that people are losing water, and because of the dehydration people are dying. So as a result of that, this was mostly noticed by Dr. Shambhunath De, and from there this oral rehydration therapy came into place. So like for example, like electoral and ORS, whatever, sometime like if we are really suffering from different types of diarrhea, then we take ORS, right, or electoral powder. So this is, there is link between this, whatever I am discussing right now. And now another important disease is smallpox, a highly contagious and deadly viral disease caused by variola virus, and often it is also called as smallpox virus. And this is very deadly disease. Like if it get in some village or in some town, then like almost all people, they are getting contaminated, like they are getting infected by this smallpox, and it is creating a lot of problem. But because of the vaccination, the disease has been eradicated, and WHO declared in 1980 that it is not there anymore. This disease itself is completely eradicated. And now there are many other infectious diseases. As I already mentioned, I am just covering few of them which are very important. So here if you see syphilis, so this is one sexually transmitted disease, and tuberculosis, this is again airborne disease. Both syphilis and tuberculosis, they are caused by some bacteria. Okay, so bacteria is the causative organism for these two diseases. In case of tuberculosis, all of you know that Mycobacterium tuberculosis is responsible for this disease. And then yellow fever, Zika and chikungunya, all these three are diseases caused by virus. And here particularly mosquito is the carrier of those viruses. So like all are viral diseases. And similarly here Ebola and AIDS, they are not carried by mosquito, but they are, I would say, Ebola is very deadly disease, and AIDS, many of you know, like this is acquired immunodeficiency syndrome caused by HIV virus, right? So that's all. Like few important, I would say, few infectious diseases I just mentioned here. But if you see, because of those infectious diseases, people used to believe, like we are thinking that microbes are always bad, like kind of common perception, because like they are creating a lot of problems, like different types of disease and so many things. But if you think, like microbes also, they are very helpful. Like as at the beginning I told, because of the microbial organism, oxygen develop in the earth, for that's one example. And then I also mentioned that Alexander Fleming, he found that penicillin from some fungus. This is also another microbes, Penicillium notatum, from there, right? Not only that, there are many microbes which are associated with human and many other organisms. They are actually helping. So that's why I am trying to say that it has some kind of complex relation between microbes and their host. For example, microbes and human. So here I will give few beneficial side of microbes. Like food production, microbes are utilized in the production of fermented food like yogurt, cheese, and bread for a very long time. So this is curd, yogurt, curd, same thing. So for example, when we we try to make curd, what we do, we just whatever curd we have, we are taking a little bit of that, just pinch of that, and we are putting that on some fresh milk, and then again that fresh milk will be converted into curd. The reason is, in that curd we had we have lot of live bacteria, mostly they are lactobacillus, and now they will make this fresh milk into curd again. So as a result of that, I'm just giving you one example. Similarly, cheese, bread, all those things, everywhere not bacteria involved, sometimes bacteria, sometimes fungus like yeast. So all those things are involved here, but for food production, they have huge role. And then digestive health. Gut bacteria, like particularly bacteria present in our mouth, in our intestine, there are a lot of bacteria present, and they break down complex food components into nutrients that can be absorbed by the body, and therefore they are playing vital role in digestion. And I must say here that healthy individual, healthy human being, we have lot of bacteria in our body, right? So the number of bacteria is more compared to the number of human cells present in our body if you are healthy enough. So sometime I mentioned that in adult human we have approximately 10 to the power 13 cells, but now I am telling in healthy human being we have much more bacterial cell compared to these 10 to the power 13 cells, because they are very small, they can still be present in our body and they are helping in different way. That's like already I mentioned digestive health. Another example is vitamin production. Certain microbes in the gut, they synthesize essential vitamins, for example vitamin K and different types of B vitamins, particularly vitamin B12. It is very important for us, and it is mostly synthesized by some bacteria present in our in our gut. And then supporting immune system, like by interacting with our immune system, beneficial microbes, they help train our immune system. So this is one positive side of this bacteria. Although those bacteria, they don't cause any disease to us, but they are helping in this way. They are giving training to our immune cells. And on top of that, if lot of these bacteria which are in a good relation with human, they are not causing disease, if those are present in our body, then some pathogen which are disease causing bacteria or disease causing organism, they cannot easily colonize inside our body. So as a result of that, they need to compete with this good bacteria present in our gut, right? So this is another strategy, I would say, it's developed over the time. So we are some kind of both are beneficial relation. Bacteria, they are surviving, and that is helping us as well. And beside all those things, there are two major points I also mentioned here. One is environmental decomposition. As you know, microbes play a crucial role in breaking down organic matter in soil, which is essential for nutrient cycling and maintaining ecosystem balance. So as a result of that, microbes such as bacteria, different types of fungus, they are the part of our ecosystem, right? And then bioremediation, certain microbes such as a few member of Bacillus and Pseudomonas, they can be used to clean up pollutants in the environment, like pesticides and oil spills. So those can be taken care of by these bacteria, which can directly clean this pesticide and oil spill. So as a result of that, if you see, microbes are also very much beneficial. They are playing very important role in our day-to-day life. And then that's all. So for more details, you can follow Prescott's Microbiology. This is a very good textbook of microbiology. Thank you very much.